Okay, so basically what I'm going to do today and the day after tomorrow would be to show some of the work we do in a very extensive manner on gene transfer, especially for cardiovascular disorders, but as you will see today, not restricted to cardiovascular disorders. And when I say gene transfer, it means genes, but it also means microRNA and SI and SH RNAs. My laboratory has developed over the last 15 years or so relatively recently, probably because I'm getting old, so I feel more the need to do that. A strong interest in cardiovascular disorders. And actually, this is really the biggest problem in the world. There is a colleague of mine in the United States who starts his seminar asking the audience, saying the audience, look at who sits on your right side, look at who sits on your left side, and then decide which of you three will die because of cardiovascular disorders. Because actually 30% of people in the world dies because of these diseases, which correspond to more than 17 million people dying every year, according to the World Health Organization. When we say cardiovascular disorders, it is in a bit less than half of the instances coronary heart disease, and in more than 1/3 to cerebral stroke, so what we call in Italy ictus. And the interesting thing, or the worrying thing, is that we tend to imagine that cardiovascular disorders are restricted to the Western world. However, the World Health Organization tells us that out of these 17 million people, 80% of people come from countries with low or middle income. And now, I mean, more people in India, for example, die because of cardiovascular disorder than for infectious disorders, and this is also the trend in the big cities in Africa. So it's really a big burden for the society, mostly due to the aging of the population. So as we tend to approach the threshold of 120 years, which is the maximum lifespan for the members of the human species, the more these kind of diseases become apparent. And if a person suffers of any kind of cardiac disease, 80% of the times this cardiac disease is ischemic, so there is atherosclerosis on the coronary arteries, and so either suddenly in a myocardial infarction or chronically in what is called ischemic cardiomyopathy, then the tissue downstream of the occlusion starts to suffer and the myocardial cell die. But there are other reasons why the myocardial cells can die. For example, if a person has hypertension for a prolonged period of time, several years, then cardiomyocytes tend to die. Or if there is a viral infection, so myocarditis, then cardiomyocytes tend to die. In any case, there is a death of myocardial cells. There is an impossibility of the heart to replenish these cells. And we will speak at length of this when we will see what we can do in the next lesson. However, the reason I'm mentioning this here is because anytime the heart cannot replenish and replicate the cell it has lost, then the heart is not unable to pump in an efficient manner and start to dilate. And this is a condition by which the pump function fails and the heart dilates, and this is known as heart failure. And heart failure is really dramatic, because once a person is diagnosed with heart failure, it has 50% of chance of not being alive only after 4 years from diagnosis. So it has a prognosis which is much worse than most types of cancer. And if you look at what's available to treat heart failure in the medical domain, so which are the medicines that are available, this is a list taken from the web. It's taken from the Mayo Clinic. The Mayo Clinic is one of the best clinical institutions in the world. So this is the list is taken, meaning to say this is the standard care for patients with heart failure. But this is not by but no means different of what you can find in Trieste, New York, Rome, or London, or any good place, clinical place in the world. So there are these 6 classes of drugs that can be used. And I spend some time in simply in going to PubMed and searching when these drugs were first introduced in the medical literature, to mean when these drugs were developed. And so, for example, you have digitalis and diuretics, where you can find some publication in 1910s or 1920s. There are anecdotal reports that, historical reports, recollection that even the Egyptians were using digitalis against this kind of disease. Then another class of drugs are the aldosterone antagonists, which were introduced in the 60s. In the 70s, 2 drugs, the angiotensin-converting enzyme inhibitors and the beta blockers. These 2 drugs really change the natural history of the disease. We provided a good treatment. And the last class of drugs in the 90s are the sartans, which means that a person who is admitted to the Mayo Clinic or Trieste, New York, London now for the treatment of heart failure is basically treated exactly with the same classes of drugs that he would have been treated 20 years ago. So it is 20 years that neither the academy nor the pharmaceutical industries have been able to develop a new drug for these conditions, a condition that is a major cause of death. 15 million people die because of this every year, and which has a prognosis by which a person who is diagnosed with heart failure has 50% of chance of not being alive only after 4 years. So the situation is really very dramatic, and over the last 20 years, being a number of treatments, this is just a partial list of drugs which have completely failed phase 3 clinical trials. Most of the big pharma companies have withdrawn research in this area because of the inability of finding new kind of compounds. If you think that this is peculiar to heart failure, you would be wrong, because this is exactly what happens in a number of other degenerative disorders. For example, in Europe, 30% of people after aged 80 or more develop a dementia, which in most cases is Alzheimer's disease. 1 person out of 3 after 80 years has Alzheimer's in Europe. Treatment for Alzheimer's, 0. Have no drug at all. 170 million people worldwide have diabetes. Treatment to re-establish beta cells in the pancreas. Beta cells are the cells that produce insulin, 0. No possibility. 30% of people over 75 are affected by retinal degeneration. So the retinal cells are lost, and this person are blind. Possibility of regenerating the retina, 0. Have nothing. Same thing for hearing. More than 50% of people after 75 don't hear well because the neuroepithelial cells in the inner ear are lost, and we have no possibility of replenishing them. So basically, what the modern medicine is hitting is a condition or a series of conditions which is difficult to distinguish from what you would call normal aging. It could be an accelerated aging for some organs. But nevertheless, these are conditions in which critical cells in some critical organs are progressively lost, and there is no way of replenishing them. So as we increase our lifespan and we approach this fatidic limit of 120 years, this condition become more and more apparent, and the big part of our medicine now and an even bigger part in the incoming years will be devoted to finding some new kind of treatments for these kind of conditions. So basically, modern medicine is a medicine of degenerative conditions related to aging, and these conditions are due to the incapacity to regenerate some critical organs in our body. The way I see human life or life of a mammal is the life of an organism who is endowed with a certain number of cells in some critical organs, and once these cells are progressively lost during life or acceleratedly lost but during life there is no way of replenishing them. Think only at the brain. We are born with about 22 billion neurons, and we lose 80,000 neurons every day. If you just make a simple calculation, you will see that in a lifespan of a person of 80 years, he loses about 12% of his brain content. This is simply the reason why aged people tend to think and to reason more slowly, are not so brilliant, they lose memory and so on. They simply lose neurons. And there is no possibility of replenishing these neurons. Since the issue here is to prevent cell loss or to try to regenerate the cells that have been lost, you understand that it is very difficult to think that one can find a very simple chemical molecule, so a conventional drug that could perform this job. Very difficult to think that you could give just a chemical molecule and start a very complex biological process that would allow, for example, the heart or the brain or the retina to reproduce the cardiomyocyte neurons or rods and cones in the retina. It is a bit more, and this is the reason why there are no drugs basically for this condition. It is a bit more reasonable to think that one could find some biological tools to perform this job. So try to re-establish some biological programs. For example, by injecting, this is the case of the heart, which is our interest, but the same could apply to the eye or to the brain or to the pancreas. For example, some genes that might reactivate a genetic program for regeneration, or for the prevention of further loss of cardiomyocytes, or peptides, recombinant proteins, or cells, obviously. We will see this in detail the day after tomorrow concerning the heart. So we need some biological tool that might somehow prevent further degeneration, or if one is very ambitious, to promote regeneration of that organ. This is the reason why there are so many interests in finding conditions where one could transfer to these kind of tissues some specific genes or nucleic acids that might reactivate these kind of programs. And here the problem is obviously a problem of how to transfer these kind of genes into these tissues. So it falls completely into the domain of gene therapy, which is nothing else than the transfer of nucleic acid for therapeutic purposes. This is not obviously, as you know, fiction. It is really reality. The first gene therapy clinical trial started in 1989 in Bethesda, Maryland, for an inherited disorders. There are now more than 1,600 clinical trials with tens of thousands of people who are being injected with nucleic acids as a drug. And when I say nucleic acids, I mean not only cDNAs coding for proteins, but also nucleic acids with regulatory purposes. For example, small DNA oligonucleotides, or siRNAs, or microRNAs, or antisense RNAs. You also see here that most of these trials, starting from 1989, however, have been remain stopped in phase 1 or 2. Phase 1 is a phase where you only look at the safety of the treatment. Phase 2, you just test in about 20 patients or so, the very limited number, the efficacy, and which and only 3.4% of the trial have approached phase 3, and none have approached phase 4. There is a single product which has been approved here in Europe by the European medicinal agents as a standard treatment for a rare inherited disorder. This obviously tells you of how difficult it is to develop nucleic acid-based drugs for the treatment of various diseases. This picture here instead tells you what are the conditions that have been treated by these, attempted to treat by these trials, and you see that gene therapy has been mostly directed so far to cancer, so as an innovative way to treat cancer, very unsuccessful in this kind of domain. Has been much more successful for monogenic disorders, so a sort of replacement therapy. There is a patient with both allele of a gene which is defective, well, I can try to cure this patient by transferring the correct allele. And there are 3 or 4 conditions, hereditary conditions, where this has been really very effective. It has become the cure for these diseases. And you also see here that there have been 9% of the trials for cardiovascular disorders, because the cardiovascular system, as you will see in a moment, is very amenable to be treated with this kind of gene, the nucleic acid-based therapy approaches. This slide here instead shows the more than 1,600 clinical trials divided by the type of delivery system that have been adopted. And you can immediately see that the vast majority of the attempts have been made by the use of viral vectors. So almost 70% of the trial have made use of viral vectors. And the reason is very simple. If you take a nucleic acid at physiological pH, the backbone, phosphate sugar, has a phosphates which are negatively charged. So there is a nucleic acid is a very big polyanion. The membrane of the cell is hydrophobic and apolar, so there is no way that such a big anion can enter freely into the cell. So you must facilitate this entry. And viruses do exactly these kind of jobs. Viruses are nothing else than small objects with a coat that protects the genome, so a nucleic acid, from degradation in the extracellular environment and permit this genome to enter into the cell. So millennia of years of evolution have just evolved viruses as these very simple objects to do exactly what we want, to transfer a gene, a nucleic acid, inside a cell. And at the clinical level, these are the 4 types of viruses that have been used so far. And on the left side, you see the viruses with the viral genes depicted in green. And on the right side, you see the vectors. So how you, through genetic engineering, you transform the virus into a tool for delivering your own gene of interest. And you immediately see that the principles are very, very simple. So basically, you cut out the genes of the virus and you substitute these genes of the virus with the genes of your own interest. I don't want to go much into detail with of these viruses, except what I'm going to tell you about is concerning this vector system here, which is one of the most promising for clinical application. This is promising for the future, but still a reality. There are more than 80 clinical trials using this virus. I mean, at the beginning of the gene therapy era, when you attended the biggest meeting for gene therapy, the meeting of the American Society of Gene Therapy. At the beginning, there were 3,000, 4,000 people attending this meeting, and the session on this virus, which is called AAV, were attended by 20 people, 30 people, so a small club of aficionados. If you go, this was the early 90s, if you go to an American Gene Therapy meeting, society meeting now, you see that the meeting on the sessions on AAV are attended by 3/4 of the attendance, just to give you an idea of the importance of this virus. You will see in a moment why it is so interesting. AAV has a very unlucky name because AAV stands for Adeno-Associated Virus, but it has nothing to do with the adenovirus. Adenovirus is a virus that is one of the common causes of the common winter flu. There are more than 80 serotypes of adenovirus diffused freely in the population. And 50 years ago, it was discovered that in a cell line which was infected by adenovirus, that the cell line was also co-infected with another kind of virus. And this is the reason why this other virus was called the Adeno-Associated Virus. But adenovirus is a very big virus. It is a double-stranded DNA virus with about 30, 35 kilobases, while AAV is a very small virus. It belongs to a family called the parvoviruses. Parvo means small. And it has a single-stranded genome, not double-stranded, of about 5 kb. And the genome is very, very simple. It has only 2 genes. They are called the rep and cap. Rep codes for some proteins which are required for the replication, and cap codes for the protein which make the shell of the virus. The virus, as I said, is very, very simple. When I speak at nanotechnology meetings, I describe this virus as the perfect bio-nanotechnological object because it has a single-strand genome and 60 proteins coming from this gene here, kappa, which makes it codes. So, and there is nothing else. So just 60 proteins that cover the virus and permits its entry, and the viral genome. We are 85% of us have antibodies against this virus, so it means that the majority of us gets in contact with this virus probably during our infancy. However, there's never been any disease associated with the virus. So it means that it is a virus that circulates in the population, however, being completely non-harmful to anybody. The virus is single-stranded except the two regions at the two extremities. These regions have about 150 nucleotides and fold back because of their primary sequence to form this sort of hairpins. And these hairpins are important because they are the start site for replication of the virus. Replication in this case is conversion from single strand to a double strand format. And normally the virus enters into a cell, and then it has two choices. If the cell is perfectly well and have no stress and is very happy as it is, the virus is converted from single strand to double strand, and then it integrates into a specific region called AAV-S1 of chromosome 19. It is the only example known in mammals of site-specific integration, so of a virus which integrates into the genome at a specific location. This occurs in about 80% of the infection, so it's relatively high. Other viruses that integrate into the genome, for example, retroviruses, you know, integrates randomly in terms of sequence preferences. Other viruses, DNA viruses, never integrates into the genome unless upon prolonged infection, some pieces get randomly integrated and usually the virus is broken. But this is the only example of site-specific integration. If the cell, however, is somehow stressed, then the virus starts to replicate. So from a single virus, a single cell can produce more than 10 to the 5, 10 to the 6 virions until the cell is completely blown up by viral replication, and this releases the virus, the virions, outside. And when I say stressed, it means that you can obtain a productive infection by a number of treatments, which include co-infection with other viruses, for example, with adenovirus, obviously, hence the name, herpes viruses, pox virus, or physical agents like radiations or treatment with a number of agents that damage DNA. So you have to stress the cells in order for the virus to replicate. This, you immediately understand, resembles what's the phase of replication of the lambda bacteriophage in bacteria. It's exactly the same. So if the cell is stressed, there is productive replication. If the cell is not stressed, there is site-specific integration. And this is the only known example in mammalian cells. Now, if you start from the viral genome, you can completely remove rep and cap and substitute these two genes with your gene of interest. So, for example, a cDNA coding for a protein you might be interested in, the expression of which is controlled by a promoter. The promoter can be a constitutive promoter, can be a tissue-specific promoter, can be an inducible promoter. And if this is a cDNA, a polydenylation site, if it is a transcript by RNA pol3, you just put the regulatory element RNA pol3 and so on. And basically, the way you produce the virus is to make this construct into a plasmid, and then you transfect cells, culture cells, EC293 cells, with this plasmid and another plasmid which contains rep and cap, but not the inverted terminal repeats, and some genes taken from adenovirus. The reason why the AAV was discovered originally is because it replicates well in cells which are stressed by adenovirus, because adenovirus has some proteins that stress the cell. And so if you put these proteins, the virus is much happier. And so the cells start replicating the virus, the virions form, eventually the cells are lysed. You can collect the virus and then purified it simply on cesium chloride gradients or by chromatography, and you obtain a very, very purified, well, depending on the way you do, you can do a simple step. They are not so purified, but they are suitable for injections in vivo into animals. And in the clinical trials, this process is made under GMP conditions, and basically you obtain something that can be directly injected into humans. Now, if you do this kind of preparation, you obtain basically a particle with a coat of 60 proteins and a nucleic acid that contain your gene of interest. Well, you take this preparation, you try to infect culture cells. For example, you can infect fibroblasts, you can take keratinocytes, epithelial cell, endothelial cell. I'm trying to remember which cell types we've tried in the past, hematopoietic stem cells, embryonic stem cells, so a lot of tumor cells in culture. The vector is absolutely awful. The worst vector you could imagine. So forget using this vector to transfer genes in culture cells. Works very, very poorly. However, if you take exactly the same preparations and you inject these directly in vivo into animals, then it is the most fantastic vectors, but only in specific tissues. And these specific tissues include, for example, the heart, but in the heart, only cardiomyocytes. The heart consists of 40% cardiomyocytes, and the rest are fibroblasts and endothelial cells. If you inject in the heart, it goes only to cardiomyocytes, but not on fibroblasts and endothelial cells. If you inject in the neurons, it goes to neurons, but preferably not to the glial cells. If you inject to the retina, basically it goes both to the retinal pigment epithelium cells and to the photoreceptors. If you inject in the skeletal muscle, it goes to the muscle fibers, but for example, not to the satellite cells. And the reason is not due to the fact that these cells lack the receptors. These vectors use very widely expressed receptors like heparin sulfate proteoglycans or sialoglycoproteins, which are expressed by most cell types. For example, in the brain, the glial cell express much more heparin sulfate proteoglycans than neurons do. However, the virus goes only into neuron. But this is due to the fact that these cells have something peculiar after internalization. And the interest for this vector comes from the fact that it won't have escaped your attention that these are exactly the same cell types which degenerate in the degenerative disorders. We spoke about heart failure, we spoke about Alzheimer, we spoke about blindness. If you inject in the retina, in the ear, it goes to the neuroepithelial cells. So basically, it has a tropism for cells that are commonly called as post-mitotic cells. So cells that have exited from the cell cycle and will never re-enter the cell cycle. Post-mitotic cells are different from non-replicating cells. For example, if you take the most common cell type in our body are endothelial cells, and the cells that line the blood vessels. In normal conditions, there are no tumors, there is no production of oocytes in the women, and there are no wounds. There is no blood vessel formation in a normal individual. And the endothelial cells are completely silent. They are completely non-replicating. They don't divide. It is estimated that in normal condition, an endothelial cell replicates every 6 months. However, this cell is still capable of replicating, and in fact, if you stimulate angiogenesis, these cells will replicate. Well, these cells are not a target for AAV. However, if you take a neuron from the cortex, for example, or a cardiomyocyte, these cells will never replicate. Whatever you do to these cells will never replicate. It is completely post-mitotic. It will live. We will see, for example, a cardiomyocyte of a neuron. There are data saying that these cells live for the whole duration of an individual. So a person who dies at 90 years have exactly the same cardiomyocyte with whom he was born, exactly the same neuron with which he was born. But this cell will never divide. And these cells are the cells that are perfect targets for AAV. Obviously, this is very, very intriguing from the molecular biology point of view. Not very much studies, I have to say, because once people in the gene therapy field started to realize this property of AAV, the most 99% of the laboratory are more interested in the applications in these tissues rather than in understanding the molecular biology. We were very puzzled by the thing. And so it's been really years that we have started side by side with our clinical, more clinical-oriented staff also to invest in understanding molecular biology. It is important, you see, for anything you do, to invest in understanding the reasons. And what we and several others observed several years ago is that you can achieve much more efficient transduction of poorly permissive cells. For example, these are hamster cells that are poorly permissive for transduction. This is an AAV expressed in GFP. You see just 2 cells. Simply by treating these cells with DNA damaging agents. For example, you treat with 3 concentration hydroxyurea, amatomycin C, or UV, you render these cells much more permissive. And in this set of experiments, we also measured the number of genomes that were found inside the cells. And you see that this is a flow cytometry measurement of fluorescence of the cells in non-permissive and permissive conditions. So after treatment with hydroxyurea, you see that there is a huge increase in permissivity by hydroxyurea. But if you measure the number of genomes that are inside the cell, you see that basically the number of genomes is exactly the same. So it means that something happens after the virus has entered into the cell. And this determines the permissivity. Remember that the virus has a single-stranded genome. And so conversion from single strand to double strand is a critical requisite for the virus to be transcribed and expressed. And one of the clue experiments that gave us some idea of what's going on is this one. So at a certain point, a few years ago, we constructed AAV vectors containing 112 repeats of a very simple DNA sequence, which is called the LAC operator. And the LAC operator in prokaryotes is the target for a protein which is called the LAC repressor. This is a regulation of LAC operon. And basically, we put 112 repeats of the LAC operator, produce an AAV vectors, and then use this to infect a poorly permissive culture cell that expressed the LAC repressor fused to GFP. So the idea was to see where the double-stranded viral DNA was accumulated inside the cell. And this was the result of these experiments. This is the time after infection. You see that instead of seeing that the LAC repressor fused to GFP became more and more fluorescent all throughout the nucleus, we started seeing the appearance of these foci of preferred accumulation of AAV DNA. This was really very, very reproducible. There are, in this cell, which is, I repeat myself, a purely permissive cell, there are about from 20 to 30 foci. They are relatively immobile in the nucleus. And we spent a couple of years in trying to understand what these foci were. We call them AAV foci. And we try to co-localize these foci with all the proteins that are known to localize in discrete compartments in the nucleus. So nucleoli, clearly these were not nucleoli. PML bodies, these were not PML bodies, or speckles for splicing, these were not speckles, and all the other compartments, until we realized that these AAV foci were exactly the same locations where proteins of the DNA damage response also accumulate. For example, if you visualize AAV foci in green and use an antibody against MBS1 or MR11 or RAD50, you see that there is a perfect co-localization of these proteins. MBS1, MR11, and RAD50 are members of a complex which is called the MRN complex. And this complex is the first protein complex that recognizes DNA double-strand breaks. There is a huge literature on this complex because it's really the complex that recognizes the double-strand breaks. And this is an antibody against an activated form, so phosphorylated on this serine, of MBS1. There is a perfect localization, so meaning also that the protein complex that is co-localized with this AAV foci is an activated complex, a complex which has been activated. Basically, MRN has been discovered because when you damage the DNA by producing double-strand breaks, you have the huge problem, the cell has a huge problem of having these breaks falling apart. And with mRNA binding to both extremities, this is a way of taking the two extremities together, and from here to signal all what's needed. For example, downstream of mRNA activation, there are ATM and ATR activation, phosphorylation of a number of other signal transduction proteins. And so basically, the cell activates DNA repair, blocks the cell cycle through activation of a checkpoint, and if repair is not successful, then the cell dies by apoptosis. So the idea here is that in a non-permissive cell, AAV enters the cell, but AAV has a single-stranded genome and hairpins at the two extremities. And these are seen as damaged DNA by the cell. So the cell recognizes this as damaged DNA, and mRNA does this job. And this prevents the single-strand genome to be converted to double-strand. Or even worse, this genome is chewed up and processed by this protein. For example, MR11 is an exonuclease that starts to chew up one strand of DNA physiologically to expose the other strand as a single-stranded, and this is the priming for homologous recombination. So it's a way when the single strand invades the sister chromatid to start homologous recombination. The idea is that a permissive cell instead is a cell where the viral DNA enters and conversion from single strand to double strand can occur, and then the viral DNA is happily there, it gets transcribed, it remains there forever. There are a number of evidences that support this DDR inhibition model, as we like to call it. For example, we know that MR11 binds AAV DNA in the cell. This is a chromatin immunoprecipitation experiment, so an antibody against MR11 in this case is able to immunoprecipitate AAV DNA, so meaning that MR11 is found attached to the genome. Or if you take cells in which you have expressed this complex of proteins here from adenovirus, you have much more transduction by AAV. This is an AAV expressing beta-galactosidase. And the function of this complex of proteins, so E1B, 55K, and E406, is exactly that, to induce MR11 degradation. So induce degradation of MR11, and this boosts up the activation of AAV. For example, if you take cells which are mutants, we are taken from patients which are mutated in this pathway of DNA damage response. For example, patient with ataxia telangiectasia, or patient with Nijmegen breakage syndrome 1, which is defective in MBS1, then these cells are much more naturally permissive to AAV transduction. Or if you take an siRNA against the components of mRNA, this is an siRNA against MBS1, you boost AAV transduction. Obviously, we wanted to know whether this also holds true in vivo, because the model predicts that there are poorly permissive cells, which are all replicating cells, but also that there are cells which have exited from the cell cycle, which are naturally permissive. We wanted to see what's going on in these cells. And the result is very simple. The post-mitotic cells don't express mRNA. This is RNA levels in the heart, the skeletal muscle, or replicating cells like the bone marrow, the spleen, or a tissue which is still capable of replication like the liver. You see that the levels of expression are much, much lower. Consider that the heart has only 40% of cardiomyocytes. And you will see much better the day after tomorrow that if we take cardiomyocytes before birth or during the first day immediately after birth, then you can find almost 40% of cardiomyocytes that are capable of replication. However, if you wait until the animal, and if you wait 1 week or 10 days, a couple of weeks in humans, then cardiomyocytes completely go out of the cell cycle, will never divide again. We exploited this to see what happens of these proteins, and this is a Western blotting on mice taken at birth, or 3 days, 7 days, 14 days, up to 2 months after birth. And you see that there is a window immediately after birth, so 1 week after birth, where these proteins are expressed, and then suddenly they are switched off, and they will never be re-expressed again. And this time window is exactly the time window when the cell will exit from the cell cycle and will never re-enter the cell cycle again. Teleologically, this makes a lot of sense, because I told you before that one of the major pathways in which mRNA is involved is to prime the damaged DNA for homologous recombination. But homologous recombination only takes place in the S phase. And the cell which has exited from the cell cycle will never re-enter the S phase. So the cell turns the expression of these proteins completely down. For example, if you take an AAV vector and you inject this AAV vector at birth, you see some transduction of the heart. However, if you wait 7 days or 21 days, you inject at that time, then the levels of transduction are very, very significantly enhanced. Let me skip this. Or if you take a siRNase against MR11, RAD50, or MBS1, you see that you boost very, very much the levels of transduction of neonatal cardiomyocytes, which are still capable of replicating. You can use also other systems. For example, a very commonly used system for laboratories who study skeletal muscle are the C2, C12 cells. These are myoblasts, which can be continuously taken in culture, replicating cells in the myoblast conditions. However, if you change the medium, you put them as a differentiation medium, these cells becomes myotubes. So they start to differentiate, they go out of the cell cycle, they become multinucleated cells with several nuclei. And you see this very clearly here. This is a marker, alpha-actinin, which is used to mark myotubes. So myoblasts in non-differentiated conditions are completely negative for this marker. However, if you differentiate the cells, they start to form these elongated polynucleated cells, which are positive for the expression of this protein. If you take an AAV vector expressed in GFP, zero transduction of myoblasts, but if you wait, they become myotubes, you start to see that the myotubes become very, very permissive to AAV transduction. And even in vivo, you can take an AAV vector expressed in GFP, inject this vector in the liver. You see some low levels of transduction. The liver is an organ which is made of cells that don't replicate, but still the capacity of, they are not completely out of the cell cycle. However, if together with the vector you inject simple short interfering RNA, so just an RNA molecule together with some lipids, then you, against MR11, RAD50, or MBS1, you increase very, very significantly the capacity of transduction. We have taken this even more systematically, because in Trieste we see this better even the day after tomorrow. We have a facility for high throughput screening. So basically we have libraries corresponding to whole genome sRNAs against all the human genes, the mouse genes, and microRNAs, as you will see, and a robotic facility to do this kind of screening. And so basically, we did an experiment in which we plated in a 384 well format in a robotized manner HeLa cells in a conditions where infection with AAV gave about 10% of the cells to be transduced by the vector. And then in each well, we put 1 out of 18,000 siRNA against all the available human genes in order to knock down each gene per well. The experiment was repeated twice. There was a very good correlation between the two experiments, and we found that there were more than almost 1,000 genes that increased out of 18,000 that increased the capacity of infection of an AAV GFP vector more than fourfold. This is a logarithmic scale. And these genes were highly enriched in genes that are involved in DNA replication, recombination repair, and cell cycle. And several of the critical components of the DNA damage repair machinery were among the genes that the integrity of which were required for AAV transduction. For example, they included PCNA, which is a polymerase delta cofactor, MDC1, gamma H2AEX, RAD50, and other genes. And in another screen, we searched for the siRNAs without AAV, the siRNAs, for the capacity of the siRNAs to induce a DNA damage inside the cells, measured by the appearance of the phosphorylated form of this histone. The phosphorylated form of this histone, so gamma H2AX, is produced by the cells in response to DNA damage. So the appearance of this means that the genomic integrity has an insult that needs to be repaired. And what we discovered was that, for example, 5 out of the top 10 siRNAs that increase AAV transduction are also able to induce gamma-2AX foci. So the meaning of which is if I use an siRNA that damages DNA, then I also increase AAV transduction. So it's a strict relationship between these kind of pathways and AAV. So basically, we now know that the reason why AAV transduction is restricted, it is because of proteins of the DNA damage response machinery, and any time you in particular touch the mRNA complex which recognizes damaged DNA, then you increase AAV transduction. Apart from this kind of biology of AAV vectors, obviously we have been very, very interested in exploiting these vectors. So in Trieste, since 10 years or so, we have a facility for the production of these vectors. There are technical persons who basically all through the year, every week, they produce 2 preparations for at least 2 different kind of vectors. We produce vectors with all the kind of variation in these 60 proteins that form the coat in order to adapt them better to the heart rather than to other kind of tissues and cell types. And this is a very, very limited, you will see in a moment, very limited list of vectors. And the reason why we have been very, very interested in these vectors is not necessarily to find something that might be useful for therapy eventually, but because we like to consider these vectors as fantastic tools for investigation for these post-mitotic tissues. So for example, suppose you are interested in a gene and you want to know what is the effect of overexpressing these genes, for example, in the heart in an adult animal. How would you approach this? You would have to construct an inducible version of your transgenic, which is activatable by a tamoxifen, for example, inducible system, and then cross this gene with cross this transgenic mouse with the mouse that produces the specificity of reactivation, so a floxed system in your transgenic, which if you are very good would take at least 1 year, 1.5 years of work. Here you do this much more simply. You simply take your gene, you clone your gene inside an AAV vector. It takes 2 weeks to produce a vector, and you inject this vector in the heart. And you have exactly conceptually the same kind of result. Not only, but you can mix your vector with another vector producing another gene, and the third vector producing a third gene, and inject the cocktail. You can produce particles of AAV up to 10 to the 14 viral particles per milliliter. If you're not familiar with these numbers, consider that if you're very good, you can produce a lentiviral vector production 10 to the 9 per milliliter, and an adenoviral vector preparation at 10 to the 11 particles. So here, from 10 to the 9 to the 14, you have a concentration which is 100,000 times more concentrated. It goes perfectly into this kind of cell types. And so over the years, we have really used a lot of this vector as tools for investigation. Just to give you a glimpse of what I mean for this, just a couple of slides. We have been very much interested in vectors that are capable to drive the induction of angiogenesis. The major angiogenic factors in mammals is this factor here, which is called VEGF-A. And you can put VEGF-A inside an AAV vectors, inject in the skeletal muscle, go to muscle fibers, and This is the result you get in a rat after 3 months of continuous expression of this factor. So these are the skeletal muscle fibers. These are endothelial cells. This circle here are capillaries seen with an antibody against CD31. And this is the amount of capillaries you see after 3 months continuous expression. And what is even more remarkable is the amount of arteries. You see that these are 2 arteries in a normal condition, and these are the arteries that you can see after 3 months continuous expression. So this vector really work really very, very well for these purposes. I skip this for the sake of time. I'm seeing that I'm getting long. We are so confident on this vector that we are also thinking now, we have been thinking over the last 2 years or 2 or 3 years to do something more using this vector. So to use these vectors to screen directly for factors in vivo. This is something that has never been done so far, as we'll see in a moment, and we are very excited about this. And I'm showing these some results just to, because I think that this is very interesting, even if it is not complete. So suppose that you have a library cloned in AAV plasmids. And suppose that inside your library there are several irrelevant genes, so cDNAs, but one gene which has a beneficial effect in some system. Then from this library you produce a library of vectors, so you have your irrelevant vectors plus 1, which is good, and then you use this vector to directly inject an animal in vivo. You wait a few months, a few weeks, and then if you inject it, for example, in the heart, you ligate the coronary artery. Or if you inject in the pancreas, then you induce diabetes. Or in the retina, you induce retinal degeneration. So the idea here is that each of the vectors will go to a different cell. And when you induce the damage, these cells will die. However, if a vector has a beneficial factor, then this cell will have a selective advantage and will preferentially survive. So you wait a few weeks, and then you recover from the tissue the AAV vectors, and this time the red vector will be preferentially selected. And then you produce another library of vectors, and then you repeat the cycle 2, 3, 4 times, as many times as needed. So this is a way, basically, of selecting for factors which provide a beneficial effect without knowing anything on how the factor works. So we call this approach the FunSel approach. So an AAV-based in vivo functional selection for therapeutic factors without absolutely knowing how the factors work. If you think at the moment this, it is like selecting the neomycin gene in HeLa cells in vitro. However, you cannot do this in vivo because there are no vector system except AAV where you can do this kind of approach, because lentiviral vectors, for example, don't go well with several cell types, including skeletal muscle or cardiomyocyte or pancreatic cells. Adenoviral vectors are very inflammatory and are removed rapidly. Plasmids don't even speak about those because they are too transient. And so AAV vectors are the only way to perform this kind of approach that's never been attempted before. So Julia in the laboratory cloned 100 cDNAs from the mouse secretome. So these are all secreted factors divided into different categories. And then Francesca, who is here, showed that it is really possible to do this. So for example, she found the right conditions, the right number of vectors where you can use. We know that we can use pools from 50 to 100 different vectors at a time. She knows what must be the length of the vector that can be used, how long she can wait before recovering the vector in tissue. So a lot of standardization work. And then Francesca, Julia, and Luca, who is also here, did a preliminary experiment in which they took the 100 vector and injected them in the pancreas, and then induced degeneration of the beta cells in the pancreas using streptozotocin. This is a chemical compound that selectively kills beta cells. And this vector was spiked with a tiny amount, an equimolar amount, of IGF, a vector expressing IGF-1, because IGF-1 is known to protect beta cells from injury. So we wanted to see if the system really works in vivo. And after 15 days, basically, what they found is that there was really 15 or 30 days, there were really a very significant enrichment for IGF-1 after treatment with streptozotocin. So basically, it means that most beta cells die by the chemical, but if a cell contains a vector expressing IGF-1, this is preferentially retained. We're very happy about this, and so basically we started some screening for new factors. I'm going to show you one screen, which is very, very visual. So this is a series of vectors, and here PCR is performed spanning the insert. So every time you see a band, this is the insert, more than one insert per band, obviously. And basically, this pool of vector was injected in a series of mice. Here I've shown 2 mice, mouse 1, mouse 2, into both legs. And then in one leg, we ligated the femoral artery to induce ischemia. So the idea was to see whether ischemia selected some vectors for beneficial function. And basically, after a couple of weeks from ischemia, then they recovered the DNA from both the ischemic and non-ischemic leg of the 2 mice and the PCR. So basically, you have to compare this ladder with this ladder, this with this, and all these with the input vector. You basically, you can't appreciate any significant differences. So this material was taken, recloned to produce another batch of E-vectors. A second round of selection was performed. This time, this band started to appear here, only in the ischemic, but not on the non-ischemic, ischemic, non-ischemic. And this band was not very relevant in the input, so there was some selection. So this was cloned and submitted to another round of selection, and this time the band became really very, very big. So we selected for something that was really spectacular, and we sequenced this band. We were very excited, and we found out that this band was ghrelin. We were absolutely ignorant about ghrelin at the moment we did this, so it was completely out of our expectations. Some of you might remember that ghrelin is a small peptide which is produced by the stomach in condition of starvation. When an animal or even ourselves are hungry, then the stomach secretes this neurohormone which goes to the brain and signals the production of growth hormone on one side. And on the other side, signals the animal that he must behave in order to search for food. So it's a major signal for the absence of food, and we selected for this factor in the skeletal muscle, which was very weird. So obviously, we wanted to test if it really worked against ischemic condition. We did experiments in the heart first, and these are hearts in which we ligated the coronary artery. And this is a measurement for heart function. You see that progressively heart function after infarction progressively goes down. This is heart failure. So the heart progressively enlarges and pumps less over time. If you give an AAV expressing ghrelin, then the function of the heart is very much preserved over 3 months of observation. At the end of the experiment, you can look at the size of the infarct, and you see that there is a very strong reduction in the size of the infarct. And if you look at 2 days after the infarct, you see a very strong reduction in the number of apoptotic cells. So basically, ghrelin worked. We selected for ghrelin because after infarct, you have a very large number of cells that apoptose. However, this number is significantly reduced if the cell produces ghrelin. You can reproduce this even in cell culture. These are cells stained with calcein in green, and these are expressing a control gene. These are expressing ghrelin. If you treat the cells in order to kill the cells with isoproterenol or doxorubicin, you kill the cells. However, if the cell express ghrelin, they are survive much better. There are other screens that I can skip that are going in the laboratory, just to come to the point that we are so much confident on these kind of approaches that now we receive funding to construct a complete collection of AAV vectors corresponding to the mouse secretome. So basically, in the human genome, there are about 20,000 genes. In the mouse genome, 23,000 genes. About 1/10 of these have a signal peptide in their coding sequence, meaning that they are putatively secreted proteins. Out of these, we can clone into AAV vectors a bit less than 1,700 of these genes, and we will have a collection of 1 vector per gene. So a huge amount of work. We have now more than, I think, 1,000. Francesca, how many we have? 900, 1,000 genes already, and we hope to have those ready in a few months. And we'll have another collection corresponding to the miRNome, so all the precursor RNAs for about 1,000 different microRNAs, and to submit these to the selection of novel therapeutic factors. So this is something that I find very exciting because, as I told in the beginning, we need something new against degenerative disorders, and probably we don't want this new going through biochemistry first. So the way usually laboratory works is to dissect a biochemical pathway and then ask the question, well, might it be that my discovered gene or pathway could be useful for heart failure or neurodegeneration? And then you test this, but most of these approaches never go to the clinics and never do something useful. Here we do completely the opposite. We don't care at all at what we will find. We only want something that works. And then we will go back and see why it works. So it's a completely different way of reasoning. And I mean, we, as I was saying, are very, very excited about this. So we are searching for genes working in the heart, in the pancreas, in the retina. Francesca is also using the library to transduce mesenchymal stem cells from the bone marrow ex vivo and then inject these cells in vivo to select for genes that improve retention of these cells in the myocardium. And you want to search also for genes that control metastasis by injecting tumors expressing specific factors. Okay, so that was my last slide. I spoke about 1 hour, I think, which was the idea. And there are a number of people who I need to thank, to acknowledge for this work. I hope not to forget anybody. Serena is responsible for all the cardiovascular animal experimentation at ICGEB. Miguel runs the high-throughput screening facility. Anna did work that I will show you the day after tomorrow. Lorena runs the AAV facility. Giulia, Francesca, Luca, I already mentioned them in the FunSel project. And these are the people who are working in Michela Zotti and Marina Dapas are responsible for the AV production facility, together with Antonella Falcione. I hope I didn't forget anybody in any case. They will forgive me, I hope. Thank you very much for your attention.